Okay, great. Well, welcome everybody. Welcome Guang. Thanks for being here. This is our second work in progress of the fall semester, and we have Guang Xu, a research professor in the microbiology department at UMass Amherst, specializing in ticks and tick-borne diseases. His research currently focuses on identifying human-biting ticks in the US and conducting molecular surveys of emerging tick and mosquito-borne pathogens impacting human health. And you'll learn a lot more about his work and current research today in his talk about identification of tick species, detection of pathogens, and progress on experimental anti-tick vaccines. Thank you for being here, Guang.
Good morning, everyone, and thank you very much for being here. As Jane said, my title today is Identification of Tick Species, Detection of Pathogens, and Progress on Experimental Anti-Tick Vaccines. So this work in progress presentation has two parts. The first part just focuses on the ticks we have collected through each project. We have a big project called is Tick Control Help. This effort brings together works from our lab at the UMass Amherst, as well as collaborations across all New England, in Connecticut, Maine, Massachusetts, New Hampshire, Rhode Island, Vermont. So I would like to thank UVAC for bringing us together. And I also like to thank all the students, postdocs, and all the collaborators. So at UVAC, we are worked on several tick projects, but today I will just focus on each project. I know each state will give a detailed report or probably several papers. So instead of showing everything, I have just picked up the data that highlights the tick species and the pathogen result. So my role in each project is to identify tick species and test them for pathogens. So you collect all those important samples. Our job is to make sure that we get the accurate tick species identification and get the good, reliable pathogen data. So compared to insects, ticks are just a very, very small group. Globally, there are about 900 tick species. In the United States, we have just over 90 tick species. And I believe 49 of them are human biting ticks, including 36 hard ticks and 13 soft ticks. So I believe we have in New England, that's just based on my counting. So we only have 17 hard ticks in New England. That's just my counting. So a key step in our each project is to know exactly which tick species we are dealing with. So morphological identification of ticks can be challenging and tricky. We all know ticks are very small. Some species look very similar. And when they suck the blood, their body changed. When you pull out of the tick, sometimes the mouth part is missing, which is most important for tick identification. Moreover, each tick life stage, they need a different identification key. So for larvae, nymphs, adult male, adult female, we need separate identification keys. So I will show you several examples that highlight just how challenging tick identifications can be. So here is the first example. Primary care providers try to identify ticks. Those are doctors and nurses. They try to identify ticks. That's the study published by CDC. You can see at the baseline, only about 10% of these primary care doctors could correctly identify an engorged adult female ticks. So only 10% of those doctors can correctly identify an engorged deer tick. 46% get the dog tick right. And we only have 3 species here. I think the most easy one is the lone star tick because that's a star in the middle of the adult female. So about 58% recognize the lone star tick. You can see about 30% of them actually failed to recognize an engorged deer tick as a tick at all. But the training is important. After training, after follow the tick-borne diseases manual, only 8% failed to recognize engorged ticks as ticks. So they improve a lot, but still, as 8% of them cannot, if you go to the ER, they will tell you, okay, that's not a tick. How about us? What about us? We entomologists, we're experts. Do we do better? So this is a study led by a European group. This European study sent 11 tick species to 14 professional labs. Those tick species were collected from Western Europe and North Africa. It contains 11 tick species. There are 2 rounds, contains 22 or 25 specimens. They were sent to 14 professional labs. They're professionals, professional entomologists. So this figure shows the misidentification rate. Even then, misidentification happened for those professionals. Those figures shows the misidentification rate by tick general. So you can see different ticks, they have different misidentification rate. Demo Center had the lowest misidentification rate at around 7%. Exodus was misidentified about 14% of the time. Rhizophyllus is the highest, about 50% of the time. So you can see, so even the experts can get it wrong. So I'm an entomologist. I made a lot of mistakes. I identified some samples, accurate is not the right name. So there are 2 rounds of identifications. At the pattern of misidentification, you can see they look similar. So it's not so surprising. After all, I'm an entomologist. So we entomologists all use the same identification key. We have spent years looking at the same characters. After a while, we got a little bit stubborn. So if the identification says, well, it is that species, we will swear it is that species, even if the tick itself disagrees. So my point here is morphology itself, morphology alone cannot solve the problem. Here is another classic example. It comes from New England. This is Professor Andrew Spilsman's 1979 classic paper. It describing a new species, Exodus diamoni from Massachusetts. In current days, we don't call it Exodus diamoni, we call it Exodus scabularis. It is the dominant species, tick species, not only in Massachusetts, but in all over the New England. However, during the 1930s and 1940s, a different tick species, not Exodus diamini. It is Exodus mirus, was abundant on Nantarke and Massachusetts, but has since become very rare. By the 1970s, many Exodus scabularis nymphs, or we call it Exodus diamini nymphs, were being misidentified as Exodus mirus. So we think, okay, Exodus mirus was abundant here, so we call it Exodus mirus. This went on for decades. Researchers just assumed that they were dealing with Exodus mirus, but in fact, Exodus scabularis was spreading. So you can see tick misidentification can distort our understanding of both vector species distribution and disease risk. Like I said before, morphological or different identification keys or better microscope cannot solve this issue. To address this, we have developed a detection pipeline that combined morphological identification with molecular tools. On the molecular side, we use the multiplex Techman real-time PCR assay. This system can do two things at once. First, it will identify 17 common tick species in the United States. It also includes Exodus racenis because a lot of travelers, when they travel to Europe, they got the Exodus racenis back to the United States. The second, it will detect 25 tick-borne pathogens, including bacteria, parasites, and RNA virus. This integrated approach provides a rapid, reliable taxonomic classification combined with pathogen strains in a high-throughput workflow. So when you submit a tick, you probably don't know how we treat your sample. So I would like to use a few slides to show you our workflow. So first, we sort and identify ticks based on the morphological characters. After that, we extract both DNA and RNA. This is Jackie. He's doing a total nucleic acid extraction. Then we run the samples. We use the multiplex real-time PCR assay for species and pathogen strain. After that, we analyze the data. We share the data with you. We summarize the data and share it with you. Finally, we store the samples for future work. So if you want to sequence the whole genome, you want to screen more pathogens, so don't worry, your sample is still safe here in -80. So in this way, from a single tick, we can identify the tick species and detect multiple pathogens at the same time. So we receive a sample, we start to sort all those ticks in two categories because Exodus and non-Exodus, they transmit quite different tick-borne pathogens. So we sort ticks into Exodus and the non-Exodus category. Each tick goes into a tube, contains a 2-millimeter metal beads. Then we put the tube onto a machine called tissue-lyzer. The machine just shake it, just like use a small hammer, just to smash all the ticks in powder. So from that, we extract both DNA and RNA together, and we create a database. For Exodus and non-Exodus, because they have different pathogens, so we use a different assay for Exodus and non-Exodus ticks. Here is an example for Exodus assay. There are 3 master mixtures here in the yellow color, there's three yellow color boxes. There's three different mass mixtures. The first mass mixture just detects 4 common tick species plus an internal control. In the US, those 4 species count over 97% of human biting ticks. So by using this mass mixture, we have a chance to cut over 97% of ticks we received. So those 4 species are Ixodes capillaris in New England, and Ixodes pacificus in the West, Amblyomma americana, and Democenter variabilis. The second mass mixture just detect Borrelia species plus Borrelia at the genus level. It will detect Borrelia burgdorferi little, Borrelia miyamotoi, and Borrelia meionii, plus Borrelia marker at genus level. The third one will detect Babesia microti, Anaplasma, and Ehrlichia murris. So we run the sample, we run everything on a 96-well plate format. So this is a 96-well plate. So we will load each master mixtures onto a 96-well plate. So I use red, yellow, and the blue color to present that. So each assay will use 32 wells. So after we load the mass mixture, we're going to load our DNA extraction. So each plate holds 29 tick samples plus 3 sets of controls. So 3 sets, each set contains 3 controls. In total, we have 9 controls in each plate. So because we have 3 master mixtures, so each tick sample will be loaded 3 times. So for example, tick number 1 will be loaded onto well A1 for master mixture 1. Then next, it will be loaded to well A5 for the second mass mixture. The last, it will be loaded onto well A9 for the third master mixture. We also include 3 controls. So the 3 sets of controls, in total we have 9. So including EC, that's extraction control. So it will detect any contamination or failure during the DNA and RNA extraction step. So if this one show up signals, we know that's some kind of contamination during the extraction step. Positive control, we call this SPC, super positive control, which contains synthetic DNA for all targets. So we have 4 tick species, we have an internal control, we have 25 different pathogens. So all this should be positive for this super positive control. So it will be always positive. No NTCs, no template control, which should always be negative. If this one becomes positive, we know that's some kind of contamination during the PCR setup. Let me show you some examples. So our real-time PCR results generate amplification curves. It's like a S-shaped curve in different wavelengths. Here we can see that's just different color. Number 1 just showed our positive controls for 5 tick species. So plus an internal control. So we'll see 5 different colors, number 1, plus an internal control. So in total 5. Figure 2 just showed a tick. It picked up our internal control. It's a hard tick 18S. So we know there's some kind of a tick there, and is more specific, hybridized to a probe specific to Ixodes capillaris. So we know this tick is a tick, and it's also hybridized to Ixodes probe. So we know this one specific to Ixodes capillaris. This is a deer tick. Number 3 showed you that's a Borrelia sample. So the Borrelia genus level signal showed up, we know that's some kind of Borrelia in that sample, and is specific hybridized to a Borrelia burgdorferi sensus lato probe. So we know there's the Borrelia there, and the species is Borrelia burgdorferi sensus lato. So the last one, just number 4, just showed a co-infection tick with both Anaplasma and Babesia. I have to mention that our TechMan real-time PCR assay is specific for Babesia microtype, but for Anaplasma, it's more general. We don't know is a deer type or a human type. So for Anaplasma, it's more general. So now let me share some results. So in 2023 and 2024, we processed 6,255 ticks. So those big 3 samples, so those big 3, deer tick, Dermacentor variabilis, and Amblyomma americana, make up 95% of our collection. So you can see Ixodes scapularis alone was 76 of the data set. The remaining species, 2 Haemaphysalis, along with Ixodes cookii and Ixodes dentalis, account for only 5% of our samples. So we showed our identification pipeline. There are 2 methods, morphological data and molecular data. Overall, from this table, you can see our morphological misidentification rate was 2.7%. And if we only focus on Ixodes, for Ixodes scapularis, it was just the misidentification rate was just 0.1%. So we achieved 99.99% of accuracy for Ixodes scapularis. But that number doesn't tell you the whole story. Here is the example. Here is the analogy. Just imagine we have 10 balls, 9 yellow color and 1 red color. So I don't even take a look. I call them all yellow color. So my identification accuracy is 90%. So that's exactly happened for Ixodes scapularis. I totally missed the red ball. So that's what happened when Ixodes scapularis dominate our collections. On the other hand, once the people know that most ticks we received are Ixodes scapularis, there is a tendency to identify uncertain species as Ixodes scapularis too. So we have seen this in practice. As you may know, we don't have a good identification key for larvae ticks in New England. We have one in California, but we don't have a larvae identification key. So you can see the Amblyomma and the Dermacentor larvae we received, they all were misidentified as Ixodes scapularis. And even dentalis and cookii nymph mistaken for that. So the conclusion is yes, without molecular identification, our morphological identification showed 99.99% of accuracy for Ixodes scapularis. But I believe this number just reflects sampling bias and observers' expectations more than true morphological identification skill. So because of sampling bias and the different identification skills, high accuracy number could be misleading. So if we look at the screen, well, if we call all those ticks are exotic scapularis, we risk missing some rare species, just like the Exodus diamondite story from 40 years ago. So one of the new WIP mission is to train the next generation of entomologists. So we put together a small collection of tick species in 1-to-1 ratio and works through the key characters with the students and technicians. We found their identification accuracy improved a lot. And here I would like to thank Nolan. Nolan did a really good job leading this effort. Thank you, Nolan. Let me show you some data. Here is the 2023 data set by state. We can see Exodus scapularis nymph dominant, except in Andrews collection in Massachusetts, where dog ticks were the majority. This could reflect a different collection site, or maybe some collectors' bias, because dog ticks are easy to find. Exodus scapularis nympho and the larvae is, sometimes hard to see. This slide shows you that's a 2024 tick species data by state. We can see Asia longhorn ticks. We found Asia longhorn ticks in Massachusetts and Rhode Island in 2023, and here we found it in Rhode Island and Connecticut in 2024. This tick clearly is spreading. They settle down in Rhode Island and Connecticut. But the question is, has it been established in Cape Cod? We need to keep an eye on it, because in those 10 years, we only got 2 Asia longhorn ticks on Cape Cod, 2 nymphs. Lone Star ticks are now in Rhode Island, New Hampshire, Massachusetts, and Connecticut. Numbers are still low. But Tom got a lot of amblyomma in his site in Rhode Island, but those ticks are moving north. The population may not be fully established in New Hampshire and Maine. Another interesting species is Exodus dentatus. We found it a large number on Cape Cod and several from Connecticut and Rhode Island. We found quite a few Exodus dentatus. The more interesting thing is 19% of them carried breeding. This slide just showed some infection rate in deer ticks. We can see Borrelia burgdorferi remains the dominant pathogen with nympho. I break down life stage by nymph and adult. So the nympho infection rate, you can see, is between 17 to 25%. And if we compare the historic data, we found this Borrelia infection rate is relatively stable year by year. But if you look at the Babesia microdi, the infection rates are 7 to 13% compared to with the historical Massachusetts data 20 years ago. We can say the Babesia seems like the infection rate of Babesia seems like is increasing. Babesia, we need to focus, more on that because it's increasing. Anaplasma, maybe some of them are tick type, deer type. We don't have ability to separate them now, but compared to the historic data, it's still a little bit higher. Let me show you some so-called rare pathogens we picked up. in year 2023, we found several rickettsia positives from the ticks, rabbit tick, Haemophilus leprosoplatris from Maine. So we did a multi-locus genotype. We found actually it is not rickettsia. And we genotype that seems like it is a new spotted fever group. The infection rate is about 6.1% in the rabbit tick we collect from Maine. After we do the phylogenetic analysis, we found that this genotype is similar to the rickettsia they found from 2 patients in North California. Interestingly, this September, just a few days ago, CDC just published a paper. Guess what? The sequence of rickettsia pathogen isolated by Bergdorfer in 1961. That was the pathogen Dr. Bergdorfer isolated in 1961. The sequence matches our genotype and the California strain. So that Bergdorfer isolate was from Montana. So now this pathogen we know we call it a new species and named it as Rickettsia linea. So we now know linea probably widely spread in the United States because we found it in California, in Montana, in Maine. So we do have spotted fever group rickettsia in New England. Another thing I'd like to mention is, I think I already did that. So the dentatis, we found the Borrelia burgdorferi sensu-salado in Ixodes dentatis. The infection rate is about 19%. We need to do more study because in sauce, usually the Borrelia in Ixodes dentatis is Borrelia andersoni. Maybe this is andersoni, but we need to run a multi-locus genotype to figure out. In Rhode Island, Amblyomma americana test positive for 2 Ehrlichia species. They're both positive for Ehrlichia evenii and Ehrlichia chaffiensis. I believe this is the first time we found Ehrlichia evenii in New England. We also detect some hotspot of Borrelia longstari in Amblyomma ticks from Cape Cod. We found a very hot spot of longstar positive ticks from Cape Cod. For Poisson virus, probably it's more widely distributed than we thought before because we found it all over the New England, in Rhode Island, Maine, Massachusetts, New Hampshire, Vermont, and a small collection from Connecticut. And I would like to tell you a striking case. So 2023, Tom collected 25 nympho Ixodes ticks from one single property. I think the property ID is 18. It turned out 8 of these 25 ticks were positive for Poisson virus. So the positive rate was 32%. So it's a very striking case. It just reminds us, maybe some Poisson virus has a very hot spot, and we need to explain this result and tell how can we tell this story to the people. We need to figure out. By the end of the year, we will have more than 10,000 ticks and the pathogen data. So this opens many opportunities for research, training, and applied work. Part of my job is to dig into the data and help you to get the most of it. So here is the example. In our each project, we're testing how effective pesticides are for tick control. If a pesticide reduce the overall number of ticks, that's good. That's great. That's good. But I might ask a deeper question. Does it reduce infected ticks and uninfected ticks equally? So because a recent study led by Osfield demonstrated a little bit different things. He found that in New York, treated with MAT52, the prevalence of Borrelia burgdorferi infected ticks went down. So MAT52 is a fungus that kills ticks. So this suggests that MAT52 likely to kill Borrelia infected ticks more than uninfected ticks. So that's very interesting, but nobody knows why. So I guess some biological control agent may work the same way, like cedar oil. I don't know. I just guess maybe some biological control agent may work the same way. They just kill infected ticks more than uninfected ticks. So this kind of things, I would like to work together with you to figure it out. How can we best use the data? Let me shift our work on anti-tick vaccine. So this anti-tick vaccine is based on BM86. This is a glycoprotein in the cattle tick midgut. The BM86 is a concealed antigen, meaning the host does not see it during the feeding. So the ticks are less likely to involve immune escape. So that's the good thing about concealed anti-tick vaccine. When antibodies from a vaccinated host reach to the tick midgut, the antibody will damage the midgut membrane. So this will let the ticks, starve to death. So it leads to higher tick mortality, smaller females, and they lay fewer eggs. So BM86 already exists. It's not new. So they have two commercial products, Tick Guard in Australia and Garlic in South America, I believe in Cuba. They have shoot up to 100% reductions in Rhaphicyphilus, and they also reduce the tick size. They change the fecundity and even improve cattle weight. So, but the production rate, it changed a lot. The efficiency changed a lot from 100% to 40%. We have successfully amplified a cloned two BM86 genes, one from Exodus deer ticks and one from Rhaphicyphilus brown dog ticks. So we use the technology called RACE. It's a rapid amplification cDNA. So this is a very large gene. It has when we align that to the genome sequence, this gene has over 20 small exons spanning over 56 kb of the genome length. So very, very large. So, and another interesting thing we found about the name. So because we predict the molecular weight is 66 kb. So we call it ID66, Exodus diamoni, molecular weight 66. So we call it this gene is ID66. ID66 is polymorphic in New England ticks. You can see after change to the amino acid sequence, there are several polymorphic sites. So BMA6 has a variable efficiency with different geographic streams of cattle ticks. Maybe this this polymorphic explained why BM86 based vaccine perform so differently in different geographic region. So when we got the full-length cDNA, the first thing we did is we express ID636 use the bacteria expression system. We try to express that in the E. coli system. But unfortunately, the expression level was very low. So since the E. coli system didn't work very well, so what can we do? We change a different system. We try to express that in a various expression system. So we tried a lot of different things, but we hope we can get a better result. But the yield is still low. After March, we finally managed to get 0.2 milligram of ID66 protein at about 60% purity. That's how much we can get. We also tried the refolding test, and all things we can do is, we get a very low yield. We get some proteins, but the yield is low. I guess that's why this ID66 has not been tested on mice or different animals. I cloned it while I was a postdoc at the National Tick Collection. That was 20 years ago. But the expression level is so low. What can we do about that? So during the COVID pandemic, mRNA vaccine were developed to create a highly effective vaccine against the new virus. This really inspired us for using ID66 as the mRNA vaccine. In 2021, with the help of Dr. Rich, we reached out to Dr. Wisman and at the University of Pennsylvania. He is the father of mRNA vaccine. He was very, very kind, and he sent us 2 mRNA vaccines based on brown dog ticks and deer ticks homology of BM86. And of course, 2 years later, it wins the Nobel Prize. In 2025, we began an immunization experiment in deer using mRNA vaccine targeting ID66. This study is including 3 immunization and 4 blood collections. The protocol was approved by the UMass ICO. So I would like to say this experiment is unique in 3 ways. First, we already said that we are using mRNA vaccines based on BM86 homology. Second, rather than working with laboratory-reared mice or rabbits, we are vaccinating deer. So currently, we have 4 deer in our deer farm. Third, we are testing under natural tick exposure. We're not going to put a lab-reared tick colony on deer. We are simulating a natural environment, and we will collect and compare ticks that naturally attached to the control and vaccine-treated deer. So I think that's 3 unique things for this experiment. So far, we have finished 2 immunization and 3 blood collection. So only 1 immunization and 1 blood collection remaining. So after that, we will start to collect ticks from deer. So our goals are twofold. The first, we will measure the immune response in deer over time. Second, we will compare the tick feeding success and fertility between the control and vaccine group. We will weight ticks, measure the egg mass, and count how many larvae hatch. So I have to admit, this has been a real learning process. We are still figuring out the best way to handle deer. Last year, they were young, they were cute, but this year they're basically teenagers and a lot of trickier. But this is not just about deer. There's also a lot of we are learning from ticks too. For example, in the bottom, there's a tube. It contains adult male, adult female nymphs and the larvae. We found on deers last week. So usually we think adult female, is a major tick stage we can collect on deer. But actually, we found quite a few larvae and the nymph on deer as well. So usually when we think of deer as a host, mainly for adult female, but we got quite a few larvae and nymph on them as well. So now we're wondering, could the vaccine affect those early life stage too? Hopefully early next year, we'll get some interesting results. I will stop here for the vaccine project. I would like to thank NUVAC Center, and I would like to thank Dr. Wisman from University of Pennsylvania. They played a key role in the development of mRNA vaccine. From University of Connecticut, Professor Verandi and his student Sun Junyan, who are developing an ELISA to detect antibodies. I would like to thank Dr. Marty Furhan from Massachusetts Wildlife. It has been very helpful for blood collection. At the University of Massachusetts, Amherst, Dr. Pearson and Melina, they have made a significant contributions to maintaining our deer farm. I would like to thank Dr. Rich. He's leading the overall efforts, guiding the team and the project forward. Thank you. Thank you all.
Thank you. Very informative talk. I appreciate it. I have a few questions. The first one is that you mentioned that 8 of the 25 specimens of exotic scapulars tested positive for possum virus. That's quite high. It reminds me a couple of years ago, one of our collaborators from Mid-Atlantic states sent us a number of ticks, and they claimed or a number of isolates, and it turns out that they were not possum virus. I was wondering whether you tested or whether you sequenced them, or it is just based on the real-time PCR.
It's just based on the real-time PCR, but that's two assays. I have a confidence, and we also send some sequence to the Yale. The sequence get confirmed. So I don't know why. Usually we found the possum virus infection rate is very, very low, probably 10,000 ticks. If you're lucky, you get a one. But that's what I don't know. I even talked to Tom. maybe that's from same animal. It's dropped from the same animal,
Yeah,
but
it
we
is
don't know.
possible. Yeah, it is possible. The other thing is that when you are referring to infestation with ticks in certain states with non-native species, we have to be very careful because the active tick surveillance that quite often we use is not effective for identifying or for finding these invasive tick species and also getting a good understanding of the infestation in the areas. I just want to give you an example. In Connecticut, we only have two counties with that are heavily infested uh, with both Lone Star and long context. So over there in some areas you go, it takes maybe half an hour to collect hundreds of these two tick species. However, if you go to other counties, you may not find it. So as a result, if you are going to generalize and say that the infestation rate is this much, that may not accurately show the extent of establishment of these tick species in various parts of any state or any region.
Thank you very much. Thank you for your comments.
Thank you. And also, last question perhaps. You mentioned that you collected uh, dog ticks, but not other, or Andrew collected dog ticks. I was wondering whether there was a seasonality to it or it was collected over the course of like year.
I can answer that question. So I'm Johanna. I'm a PhD candidate in epidemiology in Andrew Lover's lab, and the dog ticks were collected in the late spring and summer.
Thank you.
Question in the chat here. I'll just read it out loud from Matt. Based on your findings, should we continue to educate the public to identify ticks, or do you think it's better to teach them to send ticks in for testing?
So this is just my 2 cents. I think we should still educate the public to identify ticks, but at certain level. Maybe like unless, educate the people know that's a tick or engorge the tick. But for what kind of species, especially for exotics or for some certain species, we do need some professional lab get involved. The bottom line is, I think it's still important to educate public to know that's a tick and maybe at genus level.
Lars, do you have a question?
Yeah, thank you, Guang. I really appreciated the presentation, including the thoughtful comments about ticks, the issues with morphology and the value of doing both to see what the morphology is telling us and the pitfalls of morphological identification. I just had a quick question, and maybe I missed it. For the anti-vaccine trial with deer, were there 2 vaccinated deer and 1 control deer at this point in time? Is that correct?
Correct.
Okay. And then we're just waiting for the adult season to take off in October to see how that impacts. So that would be after 3 immunization doses. That was, I think, what I.
Yes.
Yeah. And then will there continue to be boosters in the spring for those deer, or?
I had a conversation with Dr. Rich and Dr. Peterson. So the first thing we like to see is the ELISA result.
Right.
So if we don't have any antibodies, probably we need to figure out, whether we're going to continue. But if we do have the antibody, I think that's at least from my side, I would like to continue.
Okay. Thank you.
Thank you.
We'll just give it a couple more minutes if anyone has any more questions.
I think Bill found a lot of larvae on deer and he published a paper, right, Bill?
Yeah, this is there's been a few studies on that. We did one with roadkill, and if you because you can't get dead deer usually outside of hunting season. So we had some roadkill and we hung them, and just a few deer that we checked, but they had juveniles, and we even found some nymphs embedded. So yeah, I think that's probably more common than we often assume, but there's been a lot of people who've looked at that over the years. That was cool that you found that too.
Thank you.
That was a great talk, Juan. Thanks. I enjoyed just learning about all the results you have. It's an incredible data set. So I really appreciate that talk.
Thank you.
Dr. Rich?
I don't know if Goodars are still on. So Guang had a paper a couple of years ago where we looked at flag ticks from Martha's Vineyard, Nantucket, and Cape Cod, and we did find small pockets where there was high incidence of Powassan. Presumably, it's a bunch of ticks that fell off one host. But it's not unheard of to see aberrantly high pockets of Powassan virus.
Any more questions or comments from anyone? That was a great talk, Guang. Thank you.
Thank you.
Well, if thanks, Jeff. If there's nothing else, thank you everyone for joining us, and we will see you at the next work in progress on October 8th.